Hello everyone and welcome back. We are on part 2 of Lab 2 lecture, and in this part we're going to look at the remaining connective tissues, the special ones, cartilage, bone, and blood, and then take a quick look at muscle and nervous tissue. Now in our last video we looked at connective tissue proper. It's very different from cartilage, and cartilage is really easy to recognize because it looks like bubbles. Now structurally, what's going on here is we still have cells that are far apart from one another. As you can see here, they're embedded in a matrix. They just look a lot different and they act a little bit differently than what we see in connective tissue proper. Now, cartilage, just like dense regular tissue, is avascular. So this is tissue that's not going to heal very quickly, if at all, if it's injured. The particular cells that we see in cartilage are called chondrocytes. The root word chondro means cartilage, and they live in these hollowed spaces called lacunae. So here's a nucleus, here's my chondrocyte, and then it lives in a cavity called a lacuna. And we'll see that again with bone as well. There is a layer outside of connective tissue, outside of cartilage, called the perichondrium, right here. The perichondrium is where we have blood vessels and nerves, and how the connective tissue, for the most part, is going to get its nutrients. So it does it through diffusion. We don't have blood vessels running through the tissue itself like we see in some other connective tissues. This is also where we're going to have new cartilage form as well. So there are 3 types of cartilage, which is really nice. So on a quiz or an exam, if you see something that kind of looks like bubbles or has some smaller cells that look like tiny little bubbles, you know it's going to be one of these 3, which is a really great place to be at. Now, hyaline cartilage is a medium-sized bubble. What that means is the cells in this cartilage are of about, sort of between the other 2 in size. It is found in a lot of different places in the body, like the nose and the ends of long bones, but it is the weakest cartilage. That's because when you look at the matrix, you see how smooth it is? The collagen fibers in here, they are fine. They're not really thick and dense, so we don't see them light up in the background, which makes it a little bit weaker compared to things like fibrocartilage. So this is the one that actually, when you think of bubbles, you want to think hyaline. Now, fibrocartilage is a little different, right? This doesn't look like bubbles. Well, depending on the slide you look at, you might actually be able to see some of the lacunae a little bit better. But one of the ways to remember what fibrocartilage looks like is to look at the matrix. It looks like brush strokes. These collagen fibers are so thick, they look like brush strokes. So it's like a beautiful watercolor in the background. Sometimes the watercolor is pink, sometimes it's like a purple, and sometimes it's blue. It just depends on the stain, but it really has nothing to do with the structural under bit of the actual matrix. So it's just a coloration. Don't let color give you anxiety. Just know it can be pink, it can be blue, it can be purple, but it looks like a really pretty watercolor. Now, because these are the collagen fibers that are making it look like that, it's because they're so big, which is why fibrocartilage is the strongest cartilage that we have. Now, the last one we have is elastic cartilage. So if you look at these cells, these are actually very, very big cells. The ones we just looked at with fibrocartilage were very, very tiny, and hyaline was somewhere in the middle. So the nice thing about all these cartilages is they all look very different. Elastic cartilage does have that bubbly appearance, but instead of the background being smooth, it is filled with these spindly, hairy-looking elastic fibers. So once again, because this is a single fiber, it's definitely something I could ask you on a quiz or an exam. So we see these bubbles and then these dark staining fibers in the matrix, which we don't see in hyaline. Now the next 2 we're going to look at are bone and blood, and we're actually going to take a lot bigger of a dive into these when we get to their respective units. So this is sort of just an introduction. In fact, next unit we're going to be looking at bone in a lot more detail. So for this test, this upcoming exam, all you'll need to be able to know is that if you see this image, something that looks like tree rings, or something that looks like this, we're looking at bone. So these are bone. Next unit we'll get into a lot more detail about what exactly we're looking at, but for this exam, anything that looks like this, it's bone. You don't even need to tell me it's compact and spongy. Same thing goes for blood. In our fourth unit, we're going to take a much closer look at blood, learn how to identify all the cells that are present in it. But for right now, if you see red cells with some other weird looking things in it, but the matrix is smooth, there's no dark fibers running through it, this is blood. Remember, if we saw red cells and dark fibers in the background, that was going to be reticular connective tissue. So we've made it through 2 of the 4 primary tissues. The third one we're looking at is muscular tissue. So muscular tissue, there are 3 types. We have skeletal, cardiac, and smooth. And this we're going to go into detail because we just don't have the time during our muscle unit. Now, both skeletal and cardiac muscle, if you look at them very closely, you will see that they are striped. We also call that striated. It's the arrangement of these cellular components that create these dark light bands. So both skeletal and cardiac have striations. The difference is skeletal muscle is cylindrical, so when we cut it, it looks like a rectangle. Cardiac muscle wraps around the heart. It creates these, it literally like wraps around and curves. So when we look at cardiac muscle, the cells are branched. So we can see a branching here as I trace it poorly, but there's one cell that's been branched. There's another cell. So we like to describe cardiac muscle as being a branched set of cells. And these cells in cardiac muscle are stapled together here. You can see these dark vertical bands. They're called intercalated discs. I will do my best to write it here. It's also in your lab manual. Intercalated disc. So these are characteristic. If you see these dark vertical bands, you know you're looking at cardiac muscle. Smooth muscle is a little different. Smooth muscle, as the name indicates, it is smooth. There are no stripes running through it. So if we look through it here, you can see it just looks like a spindle. In fact, that's how we describe these cells. They are spindle-shaped cells. They're really skinny on one end and really skinny on the other, but they're kind of fattish in the middle. So when you see this, this is going to be our smooth muscle. It's different from something like dense regular because we don't have wavy lines. We can see kind of the start and end of cells. So you might need to take a closer look at it yourself and really compare them, but they are very different. So skeletal, cardiac, and smooth muscle. Now remember, when we are making these sections, we can cut things different ways, just like you can cut a carrot a multitude of different ways. If I were to cut the cardiac muscle or skeletal muscle and make a cross section, you can see that they still look very different from one another. So when you look at a cross section of cardiac muscle, you will see that the nuclei tend to be more centralized in the cells. Okay, we can see one here and here and here. Once again, there will be places where we don't see nuclei. That just means in this particular section of time, we missed that nucleus. It might have been a little bit further down in the tissue or a little bit further up when we were cutting it. So cardiac muscle has centralized nuclei. Skeletal muscle, however, when we look at the skeletal muscle cross-section, you can see, kind of like with adipose, the nuclei are pushed to the edge. So they're kind of pushed to one side or the other. Now, this is something I don't want you to learn immediately. This is sort of the last thing you want to learn. This is the cherry on top. This is that A-level question. So make sure you're comfortable with everything else for this unit before you start tackling this. Now, the final tissue, the final thing we're going to learn for Unit 1 is nervous tissue. We will look at more nervous tissue in our final unit when we look at a nerve cell and we look at the spinal cord. But for right now, we're just going to take a quick look at what the tissue looks like. It's very almost like really dense cobwebs, and all of these little dots that you see with the spindles coming off of them, those are called glial cells. They're basically the helpers of the stars of the show, who are our neurons. So the neurons, there's one here and here and here and here. They kind of look like simple squamous epithelium, but with tendrils coming off of them. So the neurons are the ones that are creating those electrical impulses to communicate with the body and with other neurons. And the glial cells, as once again, we'll learn all of this in our final unit, these are going to kind of help the neurons and have other roles inside the nervous system. So you just need to be able to tell me, this is nervous tissue, these are glial cells, and these are neurons. And that is it, everyone. This is our final lecture for Unit 1. I'll see you in lab.